Hey all, so we're still looking at protein synthesis D1.2, but this is going to be for HL content. And we're still going to be thinking about continuity and change, and then also same questions that we had for the SL content, but we're just going to be looking at this in more detail. So the first thing we need to be thinking about is a promoter region on the DNA. And so you can see here, this looks pretty complicated, but we only really need to know two things here, this promoter region and these regulatory elements here. And so what is a promoter region? It's a section of the DNA that's right before the gene. So here's the gene that will be transcribed. And it is going to be a thing that is connected to, so you can see this molecule here, will connect onto the promoter region, and it will start the process of transcription. And so some genes have very similar promoters, and so are expressed together, which means that they are the triggers for the transcription of those genes are going to be similarly promoted. And then we have repressors and activators. And so these are called transcription factors. We have activators. So this could be an enzyme that's going to connect to an enhancer segment on the DNA. And that would encourage more transcription. So more binding to the promoter, more transcription of the gene. It doesn't have to be endless. But it's just going to, if we need more of that protein, an activator and enhancer would work together to do that. So this is going to up and down regulate how much of the gene is being made. And then a repressor, might also be an enzyme, would connect to a silencer. It's not going to entirely silence the gene necessarily, but it will slow it down. So it would then reduce the amount of components attaching to the promoter, and then the gene would be transcribed less. So an example of this would be that testosterone, for example, binds to promoters that then transcribe genes that help in the development of male reproductive organs. And I wrote this question here, how can butterflies and moths have the same DNA but look so completely different? But the reality is butterflies and moths are two totally different species, so they don't have the same DNA. That should have said butterflies and their respective caterpillars. And so what's going on there is that obviously when a butterfly is in a caterpillar stage, certain genes are being transcribed. And that changes when they turn into butterflies. And so some become silenced and some become enhanced. And what genes are being expressed is going to be different in those different stages of that organism's life. In DNA, there are segments that are called non-coding regions, and these are regions of the DNA that do not create proteins. They don't code for polypeptides. And so sections of DNA can be transcribed that will make transfer RNA or ribosomal RNA. Those are non-coding areas because those aren't proteins, they're RNA. There are sections of the DNA that are promoters, enhancers, or silencers. And so those are not coding for proteins. Telomeres, which are these green ends on this molecule here, these are related to protecting the sequence of bases in the DNA. And as mitosis occurs, these telomeres shorten as you age. And so telomeres have the length of telomeres has been linked to the aging process, but telomeres do not code for proteins. And then also introns, which we'll look at in the next slide, but these are segments that are cut out of the messenger RNA before it actually gets translated by the ribosome. So if we're looking at messenger RNA, we would have immature messenger RNA produced initially, and that initial immature RNA is going to get modified. Sorry, I had to pause the recording there because my computer was about to die. So this immature RNA is going to get cut up and have a 5' cap and a poly A tail added onto it before it's considered mature, and then it can move out of the nucleus. So introns are going to get cut out of here, and then the exons are attached together. Some of these exons can also be removed, and if they are, then a different polypeptide would be created. So the messenger RNA, the immature version, may look the same, but then be cut differently and therefore produce different polypeptides. And so the sequence of messenger RNAs that is produced is called the transcriptome, whereas the number of proteins that are made is called the proteome. And the proteome could be bigger than the transcriptome because the messenger RNA that is initially produced ends up looking different as it gets prepared. So the 5' cap and poly A tail, those things are related to aging of the and connection. So 5' cap is going to attach to the ribosome and allow the messenger RNA to connect to the ribosome. And then the poly A tail is a sequence of adenines, about 100 to 200 bases long, and it is going to degrade over time. That might be the course of 2 days, it might be 5 minutes, might be a week, but messenger RNA, once it's prepared, can be read multiple times by multiple ribosomes and make many proteins. And so the poly A tail is actually going to indicate to the ribosome whether or not that that messenger RNA should be read, or if it should be ignored and then broken down by the cell. So an example of this immature RNA being the same would be for tryptomyosin. It's coded for by the tryptinin T gene, and it has 11 exons. But in order for it to make skeletal muscle versus smooth muscle, the introns and exons get cut differently. So same messenger RNA, immature version, but then ends up looking different in the finalized versions. And that cutting process is done by something called spliceosomes, which is made up of RNA and protein complexes. So let's talk about how translation happens in detail. And so firstly, what's going on here is we have the 2 subunits of the ribosome. So the small subunit is going to be the first action point, and we have these initiation factors. Firstly, T activating enzyme is going to attach methionine onto a molecule of transfer RNA with the anticodon UAC. So UAC is the start codon. And then that initiator transfer RNA is going to bind onto the small subunit. And in conjunction with these initiating factors, they are going to trigger the messenger RNA to bind onto the small subunit at the 5' end. So it's going to start reading towards the 3'. And the UAC on the anticodon on the tRNA is going to search for AUG, which is the start component on the messenger RNA, to start reading it. And when it finds it, it's going to hook on there and it's going to bind with hydrogen bonds. Then the large subunit is going to attach on there with an initiator in the P site. So the methionine will then be in the P site, and a new transfer RNA with an amino acid is going to join into the A site. It'll create a peptide bond between the methionine and the A site amino acid. And then the methionine is going to shift, that transfer RNA is going to shift to the E site for exiting. Once that protein is made, it's also going to go through some different modifications. It's going to have that methionine removed from the 5' end. There could be changes to side chains like phosphorylation or adding carbohydrates onto the protein to change it. It could fold into its tertiary structure. So it's going to have different interactions happening there because of things like disulfide bridges, ionic bonding, covalent bonding. Disulfide bridges are an example of covalent bonding, hydrogen bonding that causes this coiling to happen. There could be parts of the polypeptide chain that are removed. And then also different polypeptides could be combined together to make a quaternary structure. That 4-0 means quaternary. And hemoglobin is an example of a quaternary structure in action. So pre-pro-insulin to insulin, this prep process is an example of modification of the protein after it's being of the polypeptide after it's being made. So this is happening in the rough endoplasmic reticulum after the ribosome has made it, and pieces are cut off. So this is the piece that gets made, the purple, blue, green, and yellow all together. And firstly, the purple piece gets cut off. That's the N terminal. And also the C section gets cut out. So then you're left with the blue and the green left. And sulfur bridges, those are covalent bonds, form between the blue and the green sections, the B and the A chain. And then a pair of amino acids is removed off of the B chain. And then you have, this is insulin, but this is the precursor to insulin. And this here is called a proteasome. And proteins only last a short time in the body. They may become denatured, they may change structurally, or they're unneeded, and they will be broken down by proteasomes into their amino acids, so those amino acids can be used again. And there's a tag called ubiquitin that attaches onto the protein when it's no longer needed, and the proteasome will come around looking for that tag and pick that molecule up. And so the blue section of the proteasome is used for recognition of which proteins need to be broken down. The red and yellow section is for digesting that protein into its respective amino acids. And then the pink piece is used for unwinding the protein so that it can be better digested. So to summarize here, there's many types of messenger RNA, transfer, messenger, and ribosomal RNA. Messenger RNA needs to be prepared before it can leave the nucleus to make proteins, and that's called post-transcriptional modification. The promoter region is a section of the DNA that allows transcription to happen, and up or down regulation is occurring due to repressors or activators. And then proteins will also be modified before they are fully mature. And so this image here is a female mouse that licks their pups a lot or does not lick their pups a lot. And so the promoter region for relaxed pups is activated, and the gene that will keep, basically you chill, the pups chill, that gene will be activated. And so the pups will have lower cortisol levels. And then that byproduct will mean that they have lower anxiety. And they, as parents, will also have higher licking or grooming. But if the mom does not lick the pups, those pups are fine, they're not dead. But that promoter will not get activated, and then the pups will be more stressed out just generally. They'll have more cortisol levels in their blood, they'll have higher anxiety, and then they will, as parents, be less likely to lick or groom their pups. So that's it for this video. Thank you for watching.